Humans are pretty cool. We use our brain and senses to understand the world around us. We smell, see, touch, hear, taste. And even though these senses work quite well, our brain always wants to understand more. And to do that, we use all kinds of tools. We invented telescopes to study stars that are too far away to see, and microscopes to look at things that are close to us but too small for our eyes. I am one of those people that work on developing these tools, and the one I work on is, in fact, a microscope. This microscope is called light sheet microscope. And first of all, it doesn't really look like this, but more like this. What is special about this microscope, apart from the fact that it looks a bit weird, is that it can be used to reconstruct 3D images of intact living samples. Imagine you want to study the 3D structure of something, the structure of, let's say, an apple. If you look at it like this, you only see the outside, and if you want to see the inside, you would need to cut the apple in slices and look at them separately. You could do the same thing with small objects, looking at the different slices through a microscope. But what if you wanted to see the structure of something that is alive? Well, if you cut it, you kill it. And what you see will not be its real living structure anymore, or how it develops with time. If the sample is transparent enough for the light to go through it, you could see its inside without cutting it. You put it under a microscope and simply focus on a plane inside it. Sadly, the image that you obtain this way is blurred by all the features that are out of focus around the plane you want to look at. Light sheet microscopes offer a great solution to this problem. A light sheet microscope uses a thin sheet of light of a specific color to illuminate a single plane inside the sample. The sample only needs to be genetically modified to express fluorescence on the parts of it we want to study. A camera in front of the light sheet looks at the fluorescence excited on an illuminated plane. One can then move the light sheet or the sample and take one image on each plane. Combining these images, we obtain a 3D image of the intact living structure of the sample. Light sheet microscopes are used to study, for example, living cells to see how they grow, move, and interact with each other. Imaging small transparent animals, like fish embryos, can help understand how their different organs develop and function. And this information can then be used to better understand our very own body. With my PhD, I help improving light sheet microscopes to make them faster and also less harmful for the samples, so that we can keep understanding more and more about our body and the world around us.